Problem formulation. What is the effect of water salinity on the respiration rate of saltwater fish and freshwater fish? The objective is to determine the effect of water salinity on respiration rate of freshwater and saltwater fish. The hypothesis: the saltwater fish will have a lower respiration rate in the freshwater, while the freshwater fish will have a higher respiration rate in the saltwater. Tools and materials. The tools and materials used in this experiment is 4 jars of water, with 2 jars of freshwater and 2 jars of saltwater. One of the saltwater jars contains the main sample of this experiment, which is the three-striped damselfish or Dasylus aruanus as a saltwater fish. And the second sample is the goldfish or Carassius auratus as the freshwater fish. A smartphone as a stopwatch, a refractometer, and a dropping pipette. Some tissue and a tea strainer. Then we move for the method. Step 1. Testing the salinity level of both water. A few drops of water is taken from the jar using a dropping pipette. The water is dropped to the surface of the refractometer prism. Close the prism with the cover slide. Wipe some excess of the water with a tissue. And then start observing. Here is the result of the freshwater salinity. The same action is repeated for the saltwater sample. And here's the result for the salt water. Step 2. Observing and counting on the respiration rate of damselfish on salt water. The operculum movement is counted in every 1 minute for 3 minutes. Step 3. Fitting the fish to a freshwater jar. The fish is taken with a tea strainer and then respiration rate is counted again for 3 minutes. Step 4. Observing and counting the respiration rate of goldfish. Step 5. Put the goldfish in a saltwater jar. The respiration rate is observed again for 3 minutes. Experimental result and discussion. Based on the experiment, here is a table to compare of what has been observed between the two specimens. As we can see here for the damselfish, when it's on the salt water with 31 ppt of salinity as their natural habitat, they have a relatively high rate of respiration. The operculum movement frequency is around 193.3 per minute. After the damselfish is put in the freshwater jar, the respiration rate slows down to 160.3 times. For the goldfish, the respiration rate is around 118 in freshwater, and it decreased drastically into 81 times when being put in the salt water. Water is always trying to reach equilibrium. Therefore, the salt moves from high concentration areas to low concentration areas. Since the ocean water is very salty, the saltwater fish will have a lower concentration of salt in their body compared to the water they swim in. As a result, most saltwater fish constantly lose water through their gills and skin. In contrast, the freshwater fish will constantly absorb water through their gills and skin. This process of controlling the flow of water across their body is called osmoregulation. For this experiment, the damselfish immediately shows a random movement in the fresh water and the respiration rate slows down because it lacks the energy for metabolism and keeping the respiration rate stable in a less concentrated water. As for the goldfish, there was a decrease in the frequency of the operculum openings at 31 ppt salinity and continuing to decrease until the 3rd minute. When the salinity is above the optimum value, the fish will try to maintain the rate of metabolism by means of osmoregulation, which requires a large amount of energy. Metabolism will only run when there is oxygen as a fuel for food. While oxygen levels are decreasing along with increasing salinity, as a result, fish will lack energy for metabolic activities, so the ability to pump water into the gills is also reduced. This situation causes the opening of the fish operculum slows down drastically. Conclusion. Based on the experiment above, we can conclude that the water salinity affects the fish respiration rate. Both saltwater and freshwater fish respiration rate decrease when being put in different water from their natural habitat. One of the hypotheses is correct, that the damselfish respiration rate is decreasing in the fresh water, but the hypothesis of goldfish increasing in salt water is proven to be wrong, as the experiment says otherwise.